grades recorded. So, and I think it's important that we get them back to you in a timely fashion. That way it's still fresh in your mind and so forth. All right. Well, let's move on and talk about the next subject. And the next subject is kind of fun stuff. So we are starting now. You've gotten some basic knowledge about proteins. You got some basic knowledge about amino acids and how things work. And what we're going to do is we're going to use that knowledge. Let's see. Let me turn this light down. If I don't turn this light down up front, then the video comes out. I get a rainbow on the screen. And so that's why I turn the light down in front. Anyway, we're going to use that knowledge to help us to do some cool things. So the title of this topic is Biotechnology. And so when we think about biotechnology, what biotechnology is applied knowledge of biology. We're using biological knowledge to make things, to do things, to create things. All right? And these are the tools that modern biochemists, modern biotechnologists use to accomplish things. And so it's important to understand some of how we accomplish doing what we do. All right? Well, as I always tell students when I first meet them, biochemistry is a business of purification. Okay? It's almost spiritual in that respect, right? So that's a joke also. But it's almost spiritual in that respect, that we're purifying. We're purifying not souls or such things, but we're purifying mostly proteins, maybe purifying nucleic acids. We could be purifying metabolites, but most of what we do is purify proteins. Okay? So a lot of what I'll talk about will be relevant to proteins, and some of it's going to be relevant to DNA and RNA, because DNA is, of course, what is used to make protein. And if I can manipulate DNA, I can manipulate the synthesis of protein. And that's a very, very powerful tool. Okay. Well, from the perspective of purification, we're really talking about separation. All right? So in this day and age, I suspect anybody who's worked in a lab probably has run a gel. How many people in here have ever run a gel? Yeah, the majority. Not everybody has, but the majority has. So what is a gel? A gel is a basic technique in separating things. Okay? What you see on the screen is a gel in which a person has separated a group of large molecules using a process called electrophoresis. And the name tells you, electro meaning electricity, and phoresis relating to movement. So we're moving things using electricity. And I said these are large molecules. These molecules might have been, in fact, in all likelihood, these molecules were nucleic acids. They could have been DNA or RNA. But we use similar techniques for separating proteins as well. And so I'm going to tell you something about how we do that. So we're talking about very large molecules. I've alluded to the fact that the largest molecules in the cell are DNAs. Right? They're way bigger than proteins are. Proteins are large, but DNA is just magnificently larger than proteins. Okay? So I'll tell you more about how electrophoresis works in a bit, but that's just an example of the kind of thing that we do. If we look at any individual band on there, that corresponds to a group of molecules, like right here where the arrow is, that are all the same size. So this might be a DNA fragment that's 1,000 nucleotides long, for example. Okay? And everything in that band would be the same DNA molecule of 1,000 nucleotides long. And it would be different from this band next to it, which might be a DNA molecule that's 1,500 base pairs long. Okay? So we can separate on the basis of size using things like electrophoresis. Okay? Well, once we separate things, we have to be able to visualize them. Being able to visualize them is important because, obviously, if we can't see them, then it makes it harder for us to work with them. So we have some techniques and tricks that we use to detect where something is that we have seen. Okay? So, for example, we use a dye called ethidium bromide, right? E-T-H-I-D-I-U-M, bromide, B-R-O-M-I-D-E. We use a dye called ethidium bromide to help us visualize fragments of DNA. So So the beauty of ethidium bromide is I can take ethidium bromide and I can take a gel like I've just run. If I took a gel and I just ran the gel and I looked at it, I wouldn't see anything. I wouldn't see those bands that I saw in that last figure. Because in visible light, DNA is essentially invisible to us. Okay? We wouldn't be able to visualize that. But if I add a dye and the dye interacts with the DNA, what the dye does is it shows me where those DNA fragments are. So those fragments, those bands that you saw in the previous gel, could have been DNAs that had been dyed with ethidium bromide. So now that ethidium bromide fluoresces and shows color at a place where there was no color previously. So that's just one way we can use to visualize something that's otherwise invisible to us. Dyes are obviously very helpful for that. Okay, well, as I'm talking about DNA, I'm going to spend the first part of what I'm going to describe to you talking about DNA. And some of the techniques we have for working with DNAs are pretty remarkable. Okay? Now, I know some of these may seem old to you, but to somebody like me who's already old, they're pretty young because they've been invented since I started working in laboratories. Okay? So the first of these is something called restriction endonucleases, or restriction enzymes. You're welcome to call them restriction enzymes since that's a little shorter name. Okay? DNA can be very large. I told you that if you took the DNA from one of your cells and you stretched all the DNA from one of your cells end to end, it would reach 7 feet. And 7 feet of DNA is not practical for you to work with. In the first place, it would fracture before you did anything, which is why when you bust open a cell, you don't see things 7 feet long springing out there. They break. Okay? Restriction enzymes allow you to break DNA into smaller pieces of specific sizes because restriction endonucleases or restriction enzymes, they cut fragments of DNA, and they cut at specific sequences of DNA. So they'll recognize a specific sequence, and they will break the phosphodiester bond at the place where it sees that sequence. Here's an example. Here's an enzyme that's very commonly used in laboratory called EcoRI. And no, you don't need to memorize the sequence, or you don't even need to memorize the enzyme. I just give it to you as an example. There are hundreds of enzymes like EcoRI, each of which recognizes a different or specific sequence. Okay? EcoRI recognizes the sequence GAATTC, going from 5' to 3'. If you read that sequence in the bottom strand, you see it also reads from 5' to 3' as GAATTC. You saw something like this one time before. Anybody remember what it was? Nobody's thought about anything since we did the last exam, right? Okay. What you saw before was a stem loop that had an inverted repeat. Remember the inverted repeat? The inverted repeat read the same way on either strand, like that. All right? So this is an inverted repeat. All right? And most restriction enzymes recognize sequences that are inverted repeats. But it's not the inverted repeat they recognize. It's actually the specific sequence, in this case, GAATTC. This guy recognizes GAATTC, and you can see it cuts between the G and the A. And down here, this one cut between the G and the A. So in each case, between the G and the A is where the cut was made, and that DNA got linearized, or got broken into pieces right there at that site. Okay? So every place if I took a human DNA and I cut it with EcoRI, what I would find is every place there was a GAATTC, it would get cut. So I get a whole bunch of different fragments, every one cut at a GAATTC. Okay? Restriction enzymes are really, really useful because I can take that sequence that I cut, and I can take another sequence that's also been cut by EcoRI, and you'll notice whenever I cut with EcoRI, I always have what we call sticky ends. What does sticky ends mean? Well, if you look at that A, it pairs with that T. And if you look at that A, it pairs with that T. And you look at that T, it pairs with that A. And you look at that T, it pairs with that A. It's sticky because they want a hydrogen bond with each other. The hydrogen bonds provide the stick. Okay? I can take another fragment that's got those exact same ends, and I can treat that whole mixture with DNA ligase. And if I do, what DNA ligase does is it sort of reverses the process. it starts putting together sequences that fit. What I could do is I could actually insert a new DNA in here, and I could make a recombinant DNA molecule. That's how you've heard of recombinants. That's how we make a recombinant. We cut DNA, and we ligate a new DNA in the middle of it. So it might be possible for me to take something I've talked about before, human growth hormone. I could take the gene coding for human growth hormone. I could cut it with a restriction enzyme, and I could put it into a bacterial DNA and put that DNA into a bacterium, and if I've done everything right, that bacterium will start making human growth hormone. That's really cool, because it means I can make something that it took a human being to make, only I can make it in the laboratory cheaply and inexpensively and give it to people who need it. Really, really powerful technique. Okay? So recombinant DNAs allow us to sort of cross those species barriers and make things that are very, very useful for us. A lot of the products that we see today are made by just what you've seen right here. A restriction enzyme and a DNA ligase. Okay. I'd like to tell the story about how restriction, or why do restriction enzymes exist. Restriction enzymes are enzymes that are found only in bacteria. They're not found in human cells. They're found in bacterial cells. And you start to think, well, why would bacteria have something that cuts up its DNA? That doesn't make any sense. Because its DNA, it wants to preserve. It doesn't want to cut up its own DNA, because if it did, it's going to die. Right? So why do they have an enzyme that'll make it die? Doesn't that mean we won't have any bacteria? Well, the reason they have restriction enzymes is obviously not a suicide mechanism or a bacterial, what do you call it? Bacterial-assisted suicide instead of a doctor-assisted suicide. By the way, the worst assisted suicide you can ever have is dentist-assisted suicide, by the way. Are there any pre-dents in here? You know, they're going to drill you to death, right? I mean, wouldn't it be awful? That's a joke. I like dentists. I like dentists. Okay. Dentist-assisted suicide. All right. Anyway, so they don't have these because they're suicidal. They have these for protection. Believe it or not, bacteria get infected by viruses just like we get infected by viruses. In fact, viruses infect every single type of cell on the face of the earth. So bacteria get invaded by viruses called bacteriophages. Or you can just call them phages, P-H-A-G-E-S. Phages are viruses that infect bacteria. They grab a hold of the outside of a bacterium, and they inject their nucleic acid into the bacterium. And that nucleic acid that goes in there has all the coding necessary to make the viral DNA and more copies of the virus, and then it explodes and goes out and infects more cells. The bacterium has a restriction enzyme to protect against the injection of that DNA. When the virus injects its DNA, the restriction enzyme says, oh, I'm going to cut this, and it cuts it into pieces, and the virus doesn't function. The restriction enzyme protects against an invading viral DNA. That part makes sense. But how about the bacterial DNA? Why doesn't it cut the bacterial DNA? Well, it turns out bacteria have a cool counter system to this. It's called a methylation system. This is the modification part of what we call restriction modification. The restriction is the cutting of the DNA. The modification relates to what you see on the screen. Let's imagine we've got a bacterium, and it's got a restriction modification system in it. It's got the enzyme to cut DNA, but it's also got another enzyme that recognizes exactly the same sequence as the cutting enzyme. So in this case, it would recognize GAATTC. And instead of cutting, this other enzyme puts a methyl group on it. It puts a methyl group on that GAATTC sequence, and the addition of that one methyl group keeps the restriction enzyme from cutting that sequence, which now means that the bacterial DNA is covered with methyl groups that keep the enzyme from cutting the bacterial DNA. So when the viral DNA comes floating in from above, it hasn't been methylated, and it gets cut. Pretty cool system. Pretty cool system. Now, at this point, there's almost always a question. Maybe this is the question right here. What is the methylation? What causes it? Okay, so our question is, what causes the methylation to happen? It's another enzyme called a methylase. Methylase. A methylase recognizes the same sequence as the restriction enzyme, but instead of cutting it, it puts methyl groups on it. The methyl groups keep the restriction enzyme from cutting it. You have a question?
So, is the reason this doesn't occur in eukaryotes because the DNA is wrapped up?
The question is, why doesn't this occur in eukaryotes? Part of it is because the DNA is wrapped up, but part of it is because we don't need it. We have an immune system. Bacteria are on their own, every cell for itself. So the protection has to be at the cellular level in the bacterium, whereas it's at the organismal level in us. Make sense? And nobody's asked the question that they usually ask, so I'll ask it for you. Well, what happens if the methylase puts a methyl group on the viral DNA that's coming in? What do you suppose would happen? It would protect it. It's exactly what would happen. The viral DNA would be protected against getting cut. And everybody goes, oh my God. And to that I say, remember, there are no perfect systems in biology. All right? In the ideal scheme, you would say, this protects it, or this would protect the cellular but not the viral. And you'd cut all the viral. And if you did that, you wouldn't have any viruses. But the fact that there are viruses tells you that there's no perfect system, right? This is a very effective system. It works very, very well, but it's not perfect. If the methylase gets there first, the viral DNA will be protected. If the restriction enzyme gets there first, the viral DNA is going to get chopped up. Yes?
Is there some mechanism that causes methylase to get there faster on a bacteria?
Okay. So the question is there a mechanism that allows it to get there faster to the bacterial DNA to protect the bacterial DNA? Yes and no. This is happening shortly after replication in bacteria. And so during replication, what's happening is you have one strand that's parent and one strand that's new, right? So that means one strand already has a methyl group on it before the other one does. That means one strand is already protected, and even if the restriction enzyme gets there first, it can only cut one of the two strands. Everything is still held together. So there's a sort of a built-in protection by virtue of the way DNA replication occurs that eventually allows the whole thing to get methylated before it gets cut. Make sense? Okay. It's not a perfect system, but it's a very good system. Well, what we do as scientists is we subvert it. We simply take, we don't want the methylase. We want to cut DNA. We don't want to protect DNA. We cut it. And we do that. So that's what we use. We use the bacterial restriction modification system. This is the equivalent in bacteria of an immune system because it's providing protection against external infection. Okay, kind of cool. All right. There are, as I said, hundreds of different enzymes cutting hundreds of different sequences. Here are some of those. No, you're not going to memorize those, the names or any of that, but I just show them to you to show that there's quite a variety of sequences that we can cut, and we do. Some recognize sequences 6 bases long. Some recognize sequences 4 bases long. This guy down here recognizes sequences that are 8 bases long, et cetera, et cetera. Okay. Cloning. I've already sort of described to you how we make a recombinant DNA. By the way, when you hear the word cloning, it has a lot of different meanings. In today's world, when you hear the word cloning, you frequently associate it with cloning an organism. cloning an animal, right? The term actually just simply means identical copies of something. So if I'm cloning an animal, I'm going to get identical animals. But people also use the term to mean cloning DNA, which means identical copies of DNA. It could mean cloning bacteria, which means identical copies of bacteria. How many people here have taken microbiology? All right? And you had to streak plates. And when you streak the plates, you got individual colonies, and every one of those colonies is a clone because it came from one cell that duplicated itself over and over and over, right? You cloned bacteria simply by streaking it out on a plate. Cloning an organism like a dog is a lot more involved. But the principle is the same idea, that you get everything that comes as an identical copy of the original. Okay? Well, what we're seeing here is making a recombinant DNA. And people use the term cloning associated with this. I tend not to use that term, but some people do. And it works like I told you. First of all, it starts with a new term for us, a plasmid. A plasmid is a very, very useful tool for a biotechnologist. A plasmid is a circular DNA that replicates in a bacterial cell. A plasmid is a circular DNA that replicates in a bacterial cell. You'll notice I said a bacterial cell. We do not have plasmids. Humans do not have plasmids. Bacteria have plasmids. Okay? You'll notice it says the word vector. Okay? A vector has other things associated with it. And with respect to molecular biology, which is what we're talking about, it has some tools associated with it. Okay? The tools that are associated with this are a couple of things. One, I said it had to replicate in a bacterial cell. What does that mean it has to have? What does replication require? It has an origin, right? Doesn't replication require an origin? So it has an origin of replication. That origin of replication has to work in a bacterial cell. Okay? So it's got an origin. It turns out it usually has something else that's very useful for us. And one of the things it has may seem a little counterintuitive to you, but I'll tell you what it is. It has within it a gene that codes for resistance to an antibiotic. A gene that codes for resistance to an antibiotic. Wow, that sounds kind of scary. Well, it's not really, right? Why does it contain a gene with resistance to an antibiotic? Well, the answer to that question relates to how we put these guys into bacteria. I'm working with a plasmid out in a test tube, and in order for me to use it, I have to put it into a bacterium. And my efficiency of putting a plasmid into a bacterium is very, very low. On a great day, it might be one cell in 10,000 that picks up the plasmid that I'm trying to give it. That's a pretty low efficiency, right? So if I have a mixture of 10 billion bacteria, and I have something that goes in at the rate of 1 in every 10,000, okay, I'm not going to have very many cells. Actually, I'll have a decent number of cells there that have it, but I have a hell of a lot more cells that don't have it. 99.99% of the cells will not get the plasmid. And I don't want them. Why do I want something? Well, let's see that's what the rest of this is about. All right? Here's that human growth hormone. I want to put this into a bacterium because I want bacteria to make human growth hormone for me. But if 99.99% of my bacteria don't have human growth hormone, I try to purify human growth hormone, my yield is going to be essentially zilch. I only want to have bacteria that have human growth hormone gene in them. It turns out I can select for them by treating the bacteria that I give the plasmid to with an antibiotic. Those that are resistant to the antibiotic will, by definition, have the plasmid. Now, If something grows on antibiotic, I have a 100% success rate, not 0.0001. I have a 100% success rate. So the selection of the bacteria with the antibiotic allows me to identify those that have what I want them to have. Very cool. Now, there's one other problem I'll explain to you in a minute. This looks like it's all very nice and simple. I've already told you there's limits of getting the stuff in. There's also limits of the efficiency with which I can insert that DNA into here. I've told you I ligate it. I get this, right? But I probably have about 1 in 10,000 that look like this, and about 9,999 that these two just come back together with no insert. I want the ones that come back like this. Right? So I've told you how to find the bacteria that gets plasmid. Now I'll have to tell you how I select bacteria that have an insert in their plasmid. That's two things. Everybody understand that? Not only do I want the bacteria that have the plasmid, but I want the bacteria that have a plasmid with this in it and not empty like this up here. Okay? All right. How do I do that? All right. Oh, by the way, there's one other thing I want a vector to have. One other thing I want a vector to have. I want this human growth hormone to be made by these bacteria. What else will I need in order for this to be made by the bacteria? We've just covered this. There's a hint. What has to happen in order for me to make protein? What process has to happen? Translation. What happens before translation? Okay, so what causes transcription to occur? What? RNA. Where do I get the RNA from? How about a promoter? Doesn't a promoter say where transcription is going to occur and cause transcription to occur there? A vector will have a promoter right next to where I want my gene to be. Because it's going to tell RNA polymerase, hey, dummy, start making the RNA here, and it does that. If I don't put a promoter in there, I can put the gene in there, but the cell isn't going to make anything because it doesn't have any place to start making messenger RNA. Let's review what's in this vector. A vector has a DNA replication origin. A vector is obviously circular, like it is a plasmid. It has antibiotic resistance. And a true vector has a promoter. Okay? Now, when I've got all those things in there and I set this up, if I do this right, that bacterium is going to make that protein that I want it to make. Bingo. All right? There's the cloning part. You guys have already done the streaking, so you know what cloning is, right? Okay. All right. There's an example vector. There's the promoter for you. There's a whole bunch of different fragments on there. We're not really concerned about them, but it tells you that there's a promoter, and it tells you there's a terminator for finishing. You don't have to have a terminator, but this one has a terminator. Okay? We want to finish that sequence. We want to finish that messenger RNA appropriately. What other thing might be up here that might be useful for us? It relates to translation. Another sequence. Well, he says a cap binding site. Turns out that actually could be useful as well, but that's not what I have in mind. How about a Shine-Dalgarno sequence? Oh, yeah. Now, see what you guys hopefully know, all right, is that if you know all these things, right, you can apply the knowledge that you have into making something. Knowledge is power, right? You can make something because you know these things are needed for transcription and translation. Here's another vector, okay? What are all these things? These are places where restriction enzymes cut this plasmid. This plasmid turns out to have 2 antibiotic resistance genes. Here's one resistance to the antibiotic ampicillin. Here's one that's resistant to the antibiotic tetracycline. It's got both in there. Think that might be useful for something? Yeah, I'll tell you, it's actually kind of useful for something. I used this one a lot when I was a graduate student. It tells you how old this guy is, all right? Let's imagine I've got a gene that I can cut with BAMH1 right there, okay? And I can cut this plasmid with BAMH1, and I can take my gene that has BAMH1 ends, and I can ligate them together, right? I'm going to have a recombinant plasmid that's going to have my DNA inserted in that BAMH1 site. And inserting that DNA into that BAMH1 site is going to destroy the function of the tetracycline-resistant gene. The ampicillin-resistant gene is still intact. How could I tell which bacteria have an insert? Very easily. Those bacteria will be resistant to ampicillin, but they will be sensitive to tetracycline. Right? Because if anything that has an insert has had something put into the tetracycline gene, that's what I'm looking for, things that have an insert. Very simple, very useful. Okay? Now, this isn't commonly used anymore, but that's one way of telling if I've made the recombinant that I want. Okay? A more common tool is actually the next plasmid that I'll show you. And you don't even know the names of these. These don't really matter. Here's one called PUC19. PUC19 turns out to be really useful. Okay? Really useful. It's got something called a polylinker, which simply means it's just got a bunch of single-cut restriction enzyme sites in there. I can cut it with EcoRI, or I can cut it with BAMH1, or I can cut it with SPH1, or I can cut it, and all those sites are in the same place. And those sites are in the middle of the lac-Z gene. You saw the lac-Z gene earlier, right? What did the lac-Z gene do? Beta-galactosidase. It cut lactose in half, right? And allowed the bacterium to use lactose for an energy source. Scientists looked at this and said, maybe we can find some useful way for this to help us, all right? It turns out, they said, well, metabolizing lactose is kind of hard to see. Maybe we could make an artificial lactose-like molecule that when the beta-galactosidase acts on this artificial molecule, I might see a color change. Okay? So they got playing around in the laboratory, and they found this molecule that was man-made called X-gal, X-GAL. And it looked enough like lactose that the enzyme beta-galactosidase cut it. Okay? Well, it turns out that X-gal has a blue color. And when the beta-galactosidase cuts X-gal, the blue color disappears. It becomes white. You can tell if a cell is making beta-galactosidase by virtue of the fact that if you treat it with X-gal, the cell will turn blue. Very simple. Anybody with me so far? No, man. Day after the exam, you've got to slow down, right? It's a good question. Arrows are showing where genes are located. So this is showing this is where the beta-galactosidase gene is, this is where the ampicillin resistance gene is, and this is where the origin of replication is. Okay? These are E. coli, yeah. These are E. coli, yep. Very benign. All right, where should I back up to? Yep. If the plasma doesn't have a terminator, how does termination happen? That would have been a good exam question. If something doesn't have a terminator sequence, how does termination happen? Anybody remember? Rho. Yeah. So Rho will terminate it. All right. Well, let's look at this vector. Let's talk about this vector. All right. So here's a plasmid. Before I showed you that you could have one that has an antibiotic resistance gene and another antibiotic resistance gene, right? And if I interrupt one, then I can tell which cells have an interrupted gene by virtue of the fact they'll be sensitive to the one antibiotic but resistant to the other, right? Everybody got that? I'm doing the same thing here, only this is not an antibiotic. This is a gene. I haven't gotten to the selection part yet. In order to understand the selection part, you need to understand what this gene is doing. This gene, if I took this plasmid right here and I put it into bacterial cells, what would they be like? Well, in the first place, they would be resistant to ampicillin because they've got an intact ampicillin gene. And if I treated those cells with X-gal, they would turn blue. Because the beta-galactosidase would cut the X-gal, the beta-galactosidase would cut the X-gal and give me a blue color. Okay? With me? No? I said that backwards. I'm sorry. Did I say that? If it cuts it, it turns blue. Oh, I'm sorry. I said the wrong thing. If it cuts the X-gal, it turns blue. Sorry. Now I've actually totally confused. Quit that. Bad professor. All right. X-gal, when you cut it with beta-galactosidase, the cells turn blue. It's only when it doesn't cut that it, I'm sorry, I said that backwards. It's when it doesn't cut that it stays white. X-gal is a white compound. So when you cut it with beta-galactosidase, you get a blue color. You know, the worst thing about making a mistake in class, besides the embarrassment of making a mistake in class, is thousands of people on YouTube will now be going, Ahern's an idiot. But you have to live with that. You have to take that chance. Okay, so just so that we're quite clear, all right, when X-gal is cut by beta-galactosidase, a blue color is produced. So I can tell cells that get this plasmid by the virtue of the fact that they will be ampicillin resistant and they will produce a blue color. Okay? Now, let's say I want to take my magic human growth hormone and I want to put it into this polylinker site. This polylinker site turns out to be useful because it's right ahead of a promoter. It's right ahead of a Shindell-Garneau sequence. Right? And if we insert our gene in there, we will disturb and disrupt the beta-galactosidase. The cell will no longer make beta-galactosidase. So now let's think about if I have my material inserted in there, and I put this recombinant in there, what's the cell going to look like? What it's going to look like is as follows. It's going to be resistant to ampicillin. And when I treat that cell with X-gal, the cell is going to stay white. Because there won't be any beta-galactosidase being made because I've interrupted the gene. That tells me not only do I have an insert, but I've got an insert in front of a promoter and in front of a Shindell-Garneau sequence, and I am cooking. Now, you earlier wanted to have a cap sequence. I've got a cap sequence about right there. Now, I can give this guy lactose. I can give this guy things that say, start making a ton of this, and what will happen is this will make a ton of this messenger RNA, and will make a ton of human growth hormone. Bang. I've used nature to make something. That's kind of cool. This is at the root of biotechnology. Absolutely at the root. Questions? Yes? How would what? How would the cell get the energy to do this? Well, the thing with this cell is that I'm giving it energy by other sources. I'd give it glucose. I'd give it plenty of energy it needs, but it sees this lactose and thinks, oh, yummy, 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 and it's fooled because it can't use it. In fact, we actually use a derivative of lactose. So we use to make it, to induce the gene. So lactose bound to the repressor, right? And that's what caused the thing to start transcribing. So we use something that's like lactose that causes lac repressor to not be able to bind to it. That's what's up. Yes? There is. That's right. So there's a promoter here. This is a functional gene system. It's already got the promoter and everything. It had nothing to do with lactose. It's a totally different promoter. Yeah, good question. Other questions? Yes?
Is there any fear that any one of these genes can be taken up by a virus or
Any fear that any one of these genes can be taken up by a virus and transported into something different. Anytime you work with anything that's coded, even outside of a laboratory, that's always a possibility. Okay? These things that we work with in laboratories are really restricted in several ways. One of the ways in which they're restricted is they have a sequence in them that prevents them from being easily transmitted. That's really important because we don't want to be transmitting antibiotic resistance around the world. So when you hear about antibiotic resistance bacteria, they're not coming from the laboratory. Okay? Laboratories are also governed by rules in terms of once you make something like this, what do you do with it? Okay? So if I make a bacterium that's antibiotic resistant in the laboratory and it's a recombinant, I have to autoclave it and kill it before I can do anything else, once I'm done with it. Okay? So I'm not releasing this out to where somebody else is going to get this somewhere. That's very important. Okay? So antibiotic resistance always gives us a concern about that, but we're very careful with what we do with these. Yes? Now you got me totally confused. So if I did what? Oh, so could I? Yeah, well, I could, but then I would have nothing to select for. Because the antibiotic is allowing me to kill off all the cells that don't get that. In the case of the other plasmid that had the 2 antibiotic resistance genes, yeah, I could do that. I could have knocked out the ampicillin and then selected for tetracycline. Make sense? Yep. But there's always a but. I'm not going to release this in the wild. No way, no.
Yeah, that's what I was trying to get into combat the antibiotic resistance.
No, because putting out a non-functional gene would do nothing. It'd be like putting out just DNA that did nothing. It wouldn't stop anything. Something that's already resistant is going to be resistant whether I put out this or not. Yes? I can make insulin with this, although with insulin it's a little bit more complicated, but yes, I could use this to make insulin. Yep. The reason it's more complicated with insulin was we had to make some cuts in the protein. So I'd have to make sure that the bacteria made those same cuts in the protein, the post-translational modification. So that would be more complicated. But I could do that, yep. Okay. Let's, just before we finish, go. There's what the multiple cloning site looks like. So I said there's a whole bunch of EcoRI, a bunch of restriction sites that's in that last plasmid I showed you. You can see there's a whole bunch of them all next to each other. Big deal. All right. Here's kind of a cool thing, okay? I'm showing how I can put it into a plasmid, but somebody asked about can I put something into a virus, all right? And it turns out that sometimes we want to put a DNA into a virus. And the reason we want to put DNA into a virus, again, we're doing this in a laboratory condition, not where a virus is getting out in the world. But putting it into a laboratory virus, because a laboratory virus has a very cool ability to inject DNA into a cell. It's very effective at that. The plasmid that I showed you, where you would put a DNA into the plasmid, the larger the DNA I try to put in that plasmid, the harder it is for me to put that plasmid into bacteria. Viruses don't have that problem. So I can take a very large DNA and put it into a virus, and the virus will say, okay, no problem, I'll stick it in the cell for you, and it does it. Okay, that's one reason we would put something into a virus. Okay. There's the selection process I told you. There's the antibiotic that I described to you in words. So we pick the cells that have the phenotype that we expect. And here's what I described to you earlier with respect to which ones get an insert. It's called blue-white screening. Now you know how that works. All right? Those that have an insert will be antibiotic resistant and white. Those that didn't get an insert but got a plasmid will be antibiotic resistant and blue. And those that didn't get a plasmid will be dead. Pretty simple. Okay. Yeah, sure. So I got 3 possibilities. All right? Bacteria that don't get a plasmid, that's going to be 99.99% of them are going to be dead when I put the antibiotic, put them in the presence of the antibiotic. All right? Those that get a plasmid that didn't have an insert will still have an intact beta-galactosidase gene. So when I treat them with XGAL, they're going to turn blue. And those that got a plasmid that had an insert that interrupted the beta-galactosidase gene, when I treat them with XGAL, they're going to be white. So blue-white screening is a very powerful technique for me to use. All right. Now, I said I was going to tell you all this stuff about protein. I didn't get around to talking about protein. I think I'm going to save that for tomorrow. I don't have a song picked out. If you want, we can do a song, or we can save a song, or I can pick out a song, or whatever you guys would like to do. What would you like to do? What's that? What do you say? Extra credit. Lots of extra credit. Well, I take suggestions, but I can listen to suggestions. I don't have to take suggestions. So it's not a nice enough day, I don't think. Do you? Or I don't know. We'll save extra credit for another day.